Hey guys, this is Kendall Terry, and this is a look at cell structure, specifically the types of cells that we see in nature. So when you look up here, we think a lot of times we think of cells, we think of microscopes, stuff like that. That's why we chose this kind of background of what's going on here. So the first thing when we look at an introduction of cell structure is cell theory. Now cell theory has three main points. It's all organisms are composed of one or more cells, and this is anything that's living, really. That two, cells are the smallest living things, the basic unit of organization. So if it's alive, it's made up of a cell, whether it's a one cell or whether it's many cells like you and I. The third is that cells arise only by division of previously existing cells. Cells only come from other cells. And there's a little video link here. You can look at this Prezi later on, and you can click on that link, and it's the video that I showed in class on how cell theory was developed, the scientists that were involved, a little bit better than what I could present it. So I just gave you the link there on the screen. Next, we have kind of the tour of a cell, the different types of cells and different things like that we're going to be looking at. The first is looking at cell size. Why are cells small? Well, kind of the basis of why cells are small is due to surface area to volume ratio. So there's a couple different ways you can think about that, but what happens as cells get bigger? Well, when cells get larger, your surface area to volume ratio changes. So if you're dealing with a small cell, 1 centimeter by 1 centimeter cube, your surface area to volume ratio is 6:1. Whereas if you up it to 3 centimeters by 3 centimeters cube, all of a sudden your surface area to volume ratio is 2:1. Another way to think of this is kind of like if our classroom compared to the gymnasium. If there's a message that has to get from the door to the middle of the classroom or from the door to the middle of the gymnasium, obviously the message is going to be able to get to the classroom quicker because the surface area from the outside, the area that it could come in to the middle of the room, the volume, is much different than in the gymnasium. So it might help you think of it like that. How do we transport information? We can get information from the door to the middle of the room and back to the door a whole lot quicker when it's small. The smaller the room, the easier that is to take place. Same with cells. Cells have to be able to communicate and interact with their environment. So if the surface area to volume ratio is like it is here with the small cube, a 6:1 or in that area, you can see that what can happen on the outside to get information in is a whole lot quicker than if we up the size. Then stuff is happening on the outside and the information being processed would be much slower. And when you're living in a changing environment like we do in nature, that is bad to take a long time then. So we want that to be a quick process. That way that cell can quickly respond to the changing environment and thus survive. So there we have why cells then are the size that they are. The larger the cell, there's kind of a point of the information just not being able to be communicated properly in an environment. So that kept cells then small evolutionarily. What are some things that all cells have? If we're kind of generally speaking about cells here, they have all cells, living cells, contain DNA. And that's everything from the prokaryote bacteria cells all the way up to the most advanced eukaryotic organisms and their cells. We have DNA in those cells. Now these are cells that are maturing cells. There are some cells that at maturity will lose their DNA, let's say, when they're functioning. Like your red blood cells, they'll lose their nucleus completely. So it's not all cells as they're in their adult state have DNA, but as they're being developed, they have to have that DNA. Another is cytoplasm, sometimes referred to as the cytosol, the cell membrane, and then the ribosomes. Ribosomes sometimes are confused, and they're included as organelles, but ribosomes are not organelles. Ribosomes are actually two subunits of RNA that fit together, and they make proteins. So many times students will misrepresent ribosomes by saying that they're organelles, but then ribosomes are found in prokaryotic cells. Prokaryotic cells don't have organelles, so they're not membrane bound. They're just two pieces of RNA that work together. The next is looking at then some of those cell structures as we compare these. Of course, if we've got a Venn diagram, and you can make one of these in your interactive notebook, you should have one in there. Of course, the middle area would be the things that we just discussed. They have DNA, they have cytoplasm, they have a cell membrane, they have cytoskeleton, which I didn't mention on there. Sometimes that's in there as well, and they have ribosomes. So that's in the middle, but let's look at each one individual now. So we've got the prokaryote, and in the prokaryote, there's some similarities here. They lack membrane-bound nucleus and organelles. They have ribosomes and DNA that just float around in their cytoplasm. Their cell wall is very rigid and made up of peptidoglycan. That's different than cell walls of plants, although you could say that something that prokaryotes and plant cells have in common would be they have cell walls, but the stuff that makes the cell walls is a little different. And that rigid cell wall gives it a shape, and that shape is actually how we talk about prokaryotic cells. So we say that a prokaryotic cell maybe is circular, which we give it the name coccus. So like streptococcus, when we look at it under a microscope, they're circular cells. Bacillus is kind of like a peel shape. Spirochete, spirals. So that's all because of the cell wall. If they didn't have a rigid cell wall, then they wouldn't have that particular shape. Another is that some of them possess flagella to aid in movement. Not all of them have flagella, but some of them do. And the way they use that flagella helps to create a hydrogen ion concentration which can drive energy production inside the cell as well. We'll talk a little bit more about that later. Eukaryotic animal, two unique things to those would be lysosomes and centrioles. We'll talk a little bit more in detail about those. Lysosomes are, of course, your trash men of the cells. They go around and they get rid of anything that's bad for the cell or any waste. And then the centrioles are involved in mitosis and meiosis, and we'll talk much more about those later. For the eukaryotic plant, you have four things that we look at. First is chloroplast. That's actually what does photosynthesis. The central vacuole and tonoplast, that's where that water is going to be stored, as well as waste products, as well as nutrients that the cell needs. It's kind of like compartmentalized in a storage area. The cell wall, which is made up primarily of cellulose, different than the prokaryotic cell wall, but it does give that structure and support. And then the plasmodesmata, which we'll talk more about when we discuss plants, and these are channels that allow the cells to communicate with each other and pass information much quicker. It's kind of like openings that connect two plant cells together. When we're talking about cells, we can't not talk about the endosymbiosis theory. The endosymbiosis theory goes along with evolution. We've discussed this more than once already, but it's an important concept that we need to understand, and it deals with mitochondria and chloroplasts. Mitochondria and chloroplasts are unique organelles in that they have a double membrane. They're surrounded by a double membrane, which goes along with the endosymbiosis theory, that one prokaryotic cell engulfed another prokaryotic cell, and they begin to work together. Thus, symbiosis. They begin to work together, and then they stayed together. They passed this down to the next generation. So there would be a double membrane. When that engulfing took place, you get the outer membrane, and then the membrane that the organism possessed would be the inner membrane. They also contain their own DNA. Now, this DNA is not doesn't hold the information for every function that the mitochondria and chloroplasts do, but it does hold the information for a lot of their functions. And they also make their own proteins. It doesn't make all of their own proteins, but they do make some of the proteins they need to function correctly. So you've got these unique cells, these unique organelles inside of the cell, the mitochondria and the chloroplast, that seem to point towards that evolutionary process of going from prokaryotic cell to eukaryotic cell. Now, the rest of the stuff in the cell would be the endomembrane system. And so now you're looking at the nucleus around the DNA, to the ER, the endoplasmic reticulum, the rough ER and smooth ER, the Golgi apparatus, and the vesicles associated with those. So this is how we get from DNA to proteins, and how proteins then move about the cell or get out of the cell or into the cell membrane. We've discussed way more of that in class, and I would suggest that you refer to your notes there. Next would be the cytoskeleton. Now, this is one that a lot of students haven't looked at much, but it really is important to how cells function. You've got three main cytoskeletons that you can see here in the circle. The first is actin filaments. Actin filaments aid in cellular movement. This is a lot of how cells move. It's also how your muscles move, is through actin filaments. Actin and myosin working together, and they kind of hook like this, and they'll attach and pull, and it allows your muscles to contract. They also use them in crawling, which is pretty cool. Your white blood cells, whenever they go to the site where maybe there's a cut on your hand and the blood comes up, these little crawling cells will come out, and they'll come out of your body and will be looking for any bacteria, and they will literally use actin filaments to move around and make sure that they're getting any bacteria that's there. You don't want to leave any of that bad stuff to get inside of the body. And then actin filaments are also involved in pinching during cellular division, which is mitosis, and the actin filaments literally make like a belt around the cell, and they just keep squeezing in until eventually the cell splits in two. Another type of cytoskeleton are microtubules. These are involved in the movement of the chromosomes during mitosis. We'll come back to those when we talk about mitosis. And they're also involved in organization. They kind of hold everything in place, making sure everything stays where it's supposed to. Stuff doesn't just float around inside of the cell, which is what a lot of people think, but it's actually got a spot, and it's held there by microtubules. And the last is intermediate filaments, mechanical strength of the cell. This is holding everything together, and intermediate filaments will even go outside of the cell membrane and even attach to another adjoining cell and then come in, kind of like those metal bars that will go through buildings that will give that structural support and integrity to the building itself. And this is what intermediate filaments do. They're just kind of that background noise that's holding everything in place. It's giving that structural integrity, making sure that the cell stays around and in its right spot as well.